Alright folks, this is 3-3 to 3-4, and we're going to look at DNA and the nucleus and things like that. Alright, so in the nucleus of the cell, the DNA is packed. Now DNA, if you completely unwound it, would be several centimeters long. So how does it fit into a nucleus, which is a couple micrometers wide diameter? I mean, most eukaryotic cells are about 10 micrometers. Some of them are big and are like 50 to 100 micrometers. But we're talking 10 to 20 micrometers for the whole cell, and then the nucleus is a portion of the cell. So how do centimeter long DNAs fit into the nucleus? And the answer is you supercoil them, you pack them. So if you look down here, this is your double-stranded DNA molecule. What happens is you wrap the DNA around a set of histone proteins. It's actually a histone octamer. There's 8 of these: 1, 2, 3, 4, 5, 6, 7, 8. And you wrap this DNA twice around that histone octamer, twice around that histone octamer, twice around the next octamer, and so on and so forth. You do have these linker histones in between the nucleosomes. When you have DNA wrapped around a histone octamer twice, that's called a nucleosome. And nucleosomes are what we say are like beads on a string. So it does look like a bead on a string. So we have the DNA wrapped around all these histones, and so we have all these nucleosomes like bead on a string, and then we pack these beads on a string, and they're packed tightly. And then we start to coil that packing. So you can see that we coil the coils, or we start supercoiling it and packing it very tightly until you get to this region up here, which is a very heavily supercoiled region. And this is how we fit very long DNA molecules. Now, when I say they're very long, they're not like a mile long. They're centimeters long. But that's very long compared to the micrometer diameter nucleus they have to fit into. So we supercoil our DNA, and when the DNA is supercoiled and stained, you can see it as these chromosomes. So chromosomes are really nucleic acid and proteins, are they not? I mean, these histones are proteins, and the DNA is nucleic acid. So a chromosome is really a combination of nucleic acids and proteins because we supercoil our DNA around the proteins. So that's how we fit long DNA into a little nucleus. The DNA is there as a template for protein synthesis. And protein synthesis is divided into 2 phases: transcription, which is copying the deoxyribonucleic acid sequences over into a ribonucleic acid sequence. In other words, transcription is copying a DNA sequence over into an RNA sequence. And we call that copy of the DNA messenger RNA. Now remember, from the last chapter, DNA has A, G, C, and T. RNA has A, G, C, and U. So the RNA copy is almost identical to the DNA, but where there was a T in the DNA, there's now a U in the RNA. All right, transcription is divided up into 3 main stages: turning on the gene, doing the actual copying, and then processing the messenger RNA a little bit at the end. You actually have to process it just a little bit. So you got to turn on the gene. That's called gene activation. You got to do the actual copying. The RNA polymerase is the enzyme that does the actual copying. And then when you're done making your messenger RNA, it's actually called pre-messenger RNA. You got to process it a little bit to make mature messenger RNA. And that's the processing part. Then, after you get messenger RNA, that leaves the nucleus and sits on a ribosome. And now we change the nucleotide sequence into an amino acid sequence. I mean, we actually don't chemically change it. It's just the code. So we say we decode the messenger RNA. And we decode it into an amino acid sequence. So now the monomers of proteins are amino acids, and you're going to have an amino acid sequence in your protein. This occurs on the ribosomes. We read 3 nucleotides at a time, and those 3 nucleotides are called a codon. And the code is non-overlapping. So if I had an AUG, CCC, UUU, well, that means something. And I read these three first, and that means methionine. And then I read these three next. See, it's non-overlapping. I didn't read AUG and then read UGC. I read AUG, and then AUG is done. I go to the next codon, the next three, CCC. Then I go to the next three, UUU. So it's a non-overlapping code, and I read three nucleotides or a codon at a time. And that's going to be translation. This is showing you transcription. Here's my gene. You receive the signal to turn it on. Maybe it's a hormonal signal, maybe some other signal. You receive the signal to turn on this gene. Okay, what happens? An RNA polymerase comes down in and starts copying the DNA, like the GAG, TAC, etc. It starts copying these into RNA. So how does it do that? It's the same base pairing that you learned before. You learned before that A binds to T and G binds to C. That's what you learned before. But now when RNA polymerase does the copying, what's happening is if there's a T in DNA, you put an A in RNA. If there's an A in DNA, which normally would be a T if you stayed DNA, is a U in RNA. Every place you think you're putting a T, put a U because it's RNA now. If there's a C in DNA, you put a G in RNA. If there's a G in DNA, it's a C in RNA. If it's a G in DNA, it's a C in RNA. So it's still A to U, G to C. A to U, G to C. G to C, G to C, A to U, G to C. We don't say A to T, G to C anymore because there is no T's in RNA. So it's A to U, G to C. And then you get your messenger RNA strand right here. Here's your messenger RNA strand. Now that's the end of transcription. The end of transcription is your messenger RNA. That's it. The product of transcription is messenger RNA. Now what happens with that messenger RNA? What happens with this messenger RNA right here is this has to go out and sit on a ribosome and be translated. Okay, so that's what has to happen here. By the way, let's look at this. See the DNA right here? ATG. From right here to right here. ATG. AUG. Every place there's a T, there's a U. CCG. CCG. Next, AGC. Next, AGC. Next, TAA. And the RNA should be UAA. Next, UAA. See, my RNA right here looks just like my DNA right here, except for there's U's instead of T's. So all I've done is transcribe it. I've copied it over. Now translation is when the RNA sits on the ribosome and then this codon right here means a particular amino acid right there. And this codon right here means a particular amino acid right there. But you're probably saying to yourself, well, how do you keep doing it? Because I have two, but there's no room for another one. You pull this whole complex through and another one comes in. And I got a video for you that'll be better. So this is transcription, where the DNA coded for this messenger RNA right here. Here's the messenger RNA. That's transcription. The messenger RNA leaves the nucleus and sits on a ribosome. It could be a free ribosome, like you see here. That's the messenger RNA sitting on a free ribosome. Or it could be a rough ER ribosome, like you see there. That's the rough ER ribosome. After the messenger RNA sits on the free ribosome, it's translated into a protein. Usually when a protein is made on a free ribosome, that protein stays in the cytoplasm of the cell. Whereas, if the messenger RNA sits on a rough ER ribosome, a ribosome that's membrane bound, it's bound to the rough ER membrane, this protein now is destined to either be in an organelle or exocytosized. And there it goes. It's going to the Golgi. The Golgi's modifying it. The Golgi's packaging it. The Golgi sends the vesicle to the surface, and you exocytosize it. You get rid of it. So proteins made on free ribosomes stay in the cytoplasm. Proteins made in the rough ER are either part of an organelle like a lysosome, or they are exocytosized and leave the cell. And this is the collage. All right, let's look at the videos of this.
The sequence of nucleotide bases in DNA carries genetic information in units that are called genes. Structural genes encode the information for specific proteins. To create a protein, a gene must first be transcribed into a sequence of nucleotide bases in a messenger RNA molecule. The messenger RNA sequence is then translated into an amino acid sequence of a protein. This sequence of amino acids in a protein molecule determines the shape and chemical characteristics of the protein. Thus, each structural gene specifies a specific protein in the cell that carries out a specific function based on its chemical characteristics and molecular shape. This function of the specific protein gives the cell and the organism the specific trait coded by the gene.
Okay, that's in a nutshell. Transcription and translation. Let's look at another. Now this is going to be a little bit more detailed. I started simple and I'm going more detailed.
A structural gene is made up of a sequence of bases in a DNA molecule consisting of a coding region with an upstream promoter and a terminator downstream of the coding region. The transcription of a gene into a messenger RNA sequence is initiated when RNA polymerase attaches to the promoter region and forms an open complex. RNA nucleotides are then polymerized from nucleotide triphosphates to make a messenger RNA. The messenger RNA sequence is elongated as the RNA polymerase moves down the DNA molecule until the RNA polymerase reaches the terminator region. Transcription is terminated when sequences in the terminator region are encountered. This causes RNA polymerase to dissociate from the DNA molecule, and the completed transcript is released.
Okay, so that's transcription. You made your messenger RNA. Now that's got to leave the nucleus and sit on a ribosome.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of one codon and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site and the ribosome shifts down by a distance of one codon. This is followed by the two amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits and the messenger RNA and protein are released.
Okay, and that's translation in a nutshell. Okay, these are the pictures going, that video is so much better, but I'll walk through these pictures. Your messenger RNA sits on your ribosome. Now, this is only half the ribosome right now because the large ribosomal subunit hasn't come in. And by the way, the video said it was a 70S ribosome. That was actually a bacterial video. 70S ribosomes are found in bacteria. Humans have 80S ribosomes. But the mechanism is exactly the same. So your messenger RNA right here sits on your ribosome. And you have your exit site, your peptidyl site, and your aminoacyl site, your EPA site. Now, the thing is, this amino acid here doesn't recognize AUG. There's nothing about methionine that would tell it to recognize AUG and be the first amino acid. However, there's an adapter molecule called transfer RNA, tRNA, as you see there. tRNA, this particular methionine tRNA, will recognize AUG. And this particular methionine tRNA recognizes methionine. So this adapter molecule is what allows methionine AUG to code for methionine. It's what allows it, because this adapter molecule is specific for methionine and specific for AUG. So this is how this whole thing starts. You got now your larger ribosomal subunit comes in. You have your methionine already there via the adapter molecule, tRNA. And your second tRNA comes in, and now it carries its amino acid. And look at this tRNA recognizes this codon, CCG. So then that comes in, and they sit together, and because they're in close proximity, I make a peptide bond. I got a dipeptide. Two monomers were just hooked together by dehydration synthesis to make a dipeptide. If what I just said is foreign, two monomers were hooked together by dehydration synthesis, make sure you go back to chapter 2 and listen to some of those chemical reactions, because if it is foreign to you, what I just said, it means you're falling behind a little bit. And maybe you actually did the material in chapter 2, but you're starting to lose it, and you got to regain it, because we're building on that material. And we're going to build on the chapter 3 material, so I can't afford you to start forgetting everything you're reading. So let me just say this again. Because these two amino acids, which are monomers of proteins, are in close proximity, they go through dehydration synthesis and form a dipeptide, or a dimer. Now what happens then? At this point in time, you'll agree that messenger RNA is hooked to my transfer RNA. There's hydrogen bonds right here. You'll agree that the transfer RNA, this one anyway, is hooked to my dipeptide. So if the messenger RNA is pulled through in this direction, then all of this is pulled through in that direction. By the way, you might say to me, well, how do you know the ribosome doesn't march its way that way? Well, it could. Matter of fact, on free ribosomes, the ribosome moves. But on rough ER ribosomes, the messenger RNA moves, because the ribosomes can't move. They're studded into the membrane. So it doesn't really matter who's moving. They're moving relative to each other. And they move. Now, let me back up again. Would you agree that when this messenger RNA and transfer RNA and dipeptide move to the left, that this transfer RNA will now be in the E site? Well, the E site stands for exit. That transfer RNA that was in the E site right there now exits. Now that freed up the A site. That's free for another transfer RNA to come in, so it does. This transfer RNA comes in, it puts the M and P in close proximity to the S, and then a dipeptide forms by dehydration synthesis. And this just keeps on happening until you make your big completed polypeptide. And here's your completed polypeptide. All right. That's translation in a nutshell. Now here's what it's telling you. This is called the genetic code table. UUU in the RNA stands for phenylalanine amino acid. AUG in the RNA stands for methionine, and so on and so forth. But please don't memorize the genetic code table. If I want to ask you a question about which codon codes for which amino acid, I will give you the genetic code table. I'll give it to you. So don't memorize this by any means. All right, the nucleus actually controls this transcription and translation, but it does it in two different ways. First of all, there's direct control, and there's direct control and indirect control. So what the heck is direct control? Well, the nucleus either allows protein synthesis to occur or doesn't. So you either make proteins or you don't. All right, so that's direct control. Synthesis of proteins. You either make them or you don't. But you could also have indirect control. And indirect control is when you make a protein that in and of itself doesn't do something, but it catalyzes a reaction that gives you a product. So here's my gene. Here's my gene. With direct control, number one, you make, say, insulin, and you lower blood sugar. Okay? Direct control. You made insulin, you lowered blood sugar. But indirect control is when you have a gene and you make an enzyme that doesn't itself do what you're looking at, but converts a substrate to a product, and B is the product that goes and does something, like lowers blood sugar or whatever. So this is indirect control because the gene didn't directly make B. The gene made an enzyme which made B. That's indirect control. See you in the next section.